Hello, I'm Adya, and I'm presenting AushadiNet-GATv2, a graph neural network model trained from scratch that predicts drug-drug interactions for cardiovascular disease treatment and also work for common treatments. As per the World Heart Report 2025, CVD remains the leading cause of death globally with a projected 20.5 million deaths in 2025, rising to 35.6 million by 2050. And also, the CVD treatments experiences the one of the highest rate of adverse drug effect ADR primarily due to polypharmacy because heart patients typically require multiple simultaneous medications. But 53% of these ADR are potentially avoidable. So here are some scope to make medication more robust. ML models trained on massive amounts of therapeutically data may help physicians make more informed clinical decisions before prescribing drugs. There are few drug interaction checker, but mostly work on rule-based rigidity, cannot generalize to new drugs or unseen combination, and also heavy to run. So, for solution, I did some research and started by leveraging graph neural networks, specifically Graph Attention Network version 2, to solve this problem as a link prediction problem. Implemented previously developed DDI models and addressing their achievement and shortcomings.
Now let's understand its architecture in short. I formulated DDI prediction as a link prediction problem on a drug interaction graph. It extracts three complementary views of each drug: Morgan fingerprints that capture substructures, MCCS keys to encode functional groups, and physicochemical descriptors quantify properties. Then properties goes to view projection and attention fusion step, where each molecular view is projected into a unified 384-dimensional embedding space through view-specific multi-level perceptrons. With batch norm and ReLU activations, then their node embedding processed by a 4-layer GATv2 graph neural network with residual connections. This design addresses the vanishing gradient problem in deep GNNs. Then comes the edge classification and dual head prediction, where the input feature vector are fed into two separate MLP branches, say heads. Then output vectors from both heads are concatenated, which cause the prediction whether drugs interact and the specific interaction mechanism, and returns the result. Let's see the actual code, the training pipeline. Here is my Jupyter notebook running on Google Colab. First I installed the dependencies. Then some memory optimization for faster training and dealing with memory constraints. Here is the main central configurations. It defined the training hyperparameters and also some bool values. If user change the bool value, training functions will change too. Then I do loading data and data sanity check for ensuring quality training. I'm using drug bank dataset and also Hackathon CSV data. Here you can see the data statistics. DDI data have total 19,000 plus drug pair and more than 1700 molecular structure. So here the actual pipeline begin. This is the step that extract the three multi-views of each drug. You can see the each view shape. Next comes the graph construction and data splitting. I also added the data augmentation to avoid the overfitting and do better generalization. And here is the main graph neural network architecture. I'm not gonna deepen it. I also created a training model tracker to save the best model. Here is another crucial cell, the model training loop. And here I started the model training initialization. I trained model for 200 epochs, and it is the training metrics plot. Let me show you the model training evaluation report. You can see the model accuracy, which falls around 90%, sometime ups or down, depends on config settings. And below I am saving the metadata and best threshold to use during actual deployment. At the end, I also created a simple prediction function here in Notebook. So you test the model yourself on real data. You can see I test the model on 3 drugs, it gives binary prediction and its probability on each drug pairs. I also created an actual model inference using Streamlit and Coder's workspace. Let's see a quick demonstration. Now I'm in my local computer with saved training weight and metadata. Here is a quick look of my Streamlit app script. To run, I started Coder server and log in and open the workspace panel. Coder handles the all deployment dependencies. Here you can see my drug interaction apps workspace is running the Streamlit app. I enabled the patient profiling and adjusted the details and entered the drug's name and clicked predict. It predicted the each pair interaction for its ADR. It choose interaction binary decision, 1 or 0 for yes or no, and also probability of its binary decision. And then important interaction type and its probability also. Application has an OCR feature. You can upload the prescription image and it will extract the drug's name and do prediction on it. Application pull the drug description from PubChem API and also give personalized verdict.